Welcome to this video lecture program. This is Ramakrishnan, Assistant Professor from Department of Biochemistry, St.Joseph College of Arts and Science, Cuddalore. In today's program, I am going to discuss the topic denaturation and renaturation of DNA with a melting curve and hyperchromicity. DNA is the genetic material in all the living organisms. This DNA genetic material exists in a double helical structure that is made by two separate strands. The two strands are arranged in an anti-parallel direction, and the two strands are held together by hydrogen bonds, that is stabilized by hydrogen bonds in between the purine and pyrimidine nucleotides. As per Watson and Crick base pairing, adenine always pairs with thymine and guanine always pairs with cytosine. So the stability of the DNA is maintained by two hydrogen bond between the AT and three hydrogen bond between GC. Thereby the double helical structure of DNA is stabilized. So this DNA exists in native state, that is active form of DNA in double helical structure. This DNA will undergoes denaturation and loses its activity. Denaturation is the phenomenon of change in the loss of biological activity of a compound. This denaturation may cause the activity loss, thereby that compound will lose its originality. Whereas renaturation is the process of conversion of the denatured molecule into a native state. During this process, so many molecules will regain its originality or native state, but some case it may not be irreversible. For example, in denaturation of DNA, maximum case it will be irreversible, whereas in the case of DNA, this denaturation is generally a reversible mechanism. And we have to analyze another term, DNA denaturation with melting. Melting is the simple term which relates with the conversion of one state to another one in the presence of heating. For example, candle, when we heat it, that converted into a solid state into a semi-solid liquid state, that is called a melting of candle. Same way, the genetic material is converted into a denatured form, that is double helical to a single stranded DNA strand in the presence of heating, means that is called a DNA denaturation or DNA melting. In this case, the DNA solution is heated. As a result of this heating, the hydrogen bonds between the two strands gets disturbed, and the biological activity, that means the two strands get separated. But this disturbance of this hydrogen bond is brought by that heat, and the heat requirement, for example 25 degree, 30 degree, like this, the increase in temperature will be determined by the nature of the hydrogen bonds. For example, if your DNA is rich in AT base pairs, then automatically two hydrogen bonds are heavy, so the denaturation will be easier. Whereas the same DNA is rich in GC, means that is guanine, cytosine, there are three hydrogen bonds. That will take more energy to disturb these three hydrogen bonds. Therefore, its melting temperature will be high. Okay. So therefore, the DNA gets denatured because of the presence of the temperature. So it is called a DNA melting. Thus, the denaturation of DNA is the loss of helical structure from the double helical structure into a single strand. So this single strand conversion will be converted once again, regenerated into a double helix called annealing or renaturation when we remove the external temperature. Same way of simple candle. So when we reduce the external temperature, that molten state candle turned to solidified solid state. Same way, the DNA will be reorganized into original state, that is called renaturation of DNA or annealing. This is brought by temperature, change in temperature, that is called melting temperature. So how shall we determine the melting temperature of a DNA? So we will find out the melting temperature because it is an unique character for the DNA sample. For example, prokaryotic DNA with various species, or even in the eukaryotes, humans, horses, anything, each and every one will differ in their melting temperature. That is unique character for the species DNA. So it is used for identifying the nature of the DNA source, plants or animals, or what is the specific species. All these things will be analyzed based on the peculiar character called melting temperature, because all these DNAs will differ with each other based on the AT and GC content. So measuring the DNA denaturation, how far it is denatured, whether it is completely denatured or partially denatured or not at all denatured. That is original DNA, double helical stranded DNA means it is native DNA, and complete single strand DNA means denatured DNA. So in between the two, at a particular stage, the DNA, it will be in 50% denatured and 50% in native state. That is called a TM, or temperature midpoint, or melting temperature. So temperature midpoint, in that is mentioned as TM, that is the point in which the 50% of DNA is turned into single strand as well as double helical strand. So this will be measured by the kinetic study using UV absorption spectrophotometer. Generally, double helical structure of DNA will absorb the UV light at 260 nanometer. So that is called DNA absorption. So that is because of the pi electronic clouds present in the aromatic ring in A, T, G and C. That is adenine, thymine, cytosine and guanine. All these bases are present in the central part of the double helical DNA. When the DNA is under melting, this double helical strand is turned to single strand DNA. Therefore, the DNA is ready to absorb the UV light. That is DNA containing free nitrogenous bases which is facing in the side direction. Whereas in the double helical structure of DNA, all the purine and pyrimidine nucleotides are stacked in between the two strands. That is just like placed in the inner side. So when the UV light is passed through this way, all the AT and GCs are all protected by repeated phosphate pentosugar backbone. That is the basic backbone of the DNA. Therefore, the pentosugar phosphoric acid will face the UV light, and the AT GCs are all in the central part. So minimum absorption of UV light takes place at a double helical strand. Whereas in the single stranded form, all the base pairs are all free to absorb the UV light and easily absorbs and exhibit maximum absorption. So this curve, that is DNA melting curve based on UV absorption light, indicates that at the native state of DNA, that is in double helical strand, DNA will show minimum UV light absorption at 260 nanometer. When we increase the temperature, the double helical strands gets denatured and converted into a single strand and exhibit maximum UV light absorption. At a particular point, that is the temperature midpoint, so 50% of DNA gets denatured and shows a particular standard unique value for every DNA molecules. This study will help to find out the specific DNA present in the sample and to find out whether the DNA is in native state or in the denatured form. So here the technique UV absorption spectroscopy with the controlled thermal system is an essential one for this study. Therefore, we will find out the hyperchromicity. That is because of increase in the absorption during the denaturation of DNA. So it is called a hyperchromicity because increase in the absorption of DNA. Thank you.